Hello everyone, welcome back to my channel Competitive Botany. In this video we are going to see the second part of non-allelic gene interactions such as inhibitory genes, duplicate genes, polymorphic genes. These three topics are discussed in this video. Let's see the first topic, nothing but inhibitory factor or inhibitory genes. So inhibitory gene has no phenotypic effect but it inhibits the expression of another non-allelic gene. Okay, inhibitory gene is nothing but it does not produce any phenotypic effect but it inhibits the expression of other non-allelic gene. Remember, okay. So for this, leaf color in rice is the example. Let's see the example for a better understanding. So in rice plants, purple leaf color is due to the gene capital P. Capital P gene will produce purple leaf color in rice plants, whereas small p causing green color in rice plants. Another non-allelic gene is present, nothing but capital I. It has no phenotypic effect on leaf color, but it inhibits the expression of capital P. Okay, it does not produce any phenotypic effect even though it inhibits the expression of purple color. So when it is in the form of recessive form, nothing but small I, if small I is present, it is ineffective. That means it is not inhibiting the expression. It will not inhibit the expression. Okay, so thus the factor I has no visible effect, of course, but it inhibits the expression of capital P. That means purple leaf color is inhibited by capital I gene. Okay, so let's see the cross. Here the pure breed purple leaf colored rice plant is crossed with a pure breed green leaf colored plant. These are used as a parents when they produce gametes during the course of reproduction. Okay, so the F1 hybrid will be produced. Here both the capital genes, capital P is present and capital I also present. That's why it is producing the green color leaves. Why? Because capital I is present. It is suppressing the expression of capital P. Okay, so that's why the F1 individuals are in green color. Okay, when they are allowed to self-pollination, we are going to get F2 progeny. Let's see the F2 punnet board. So these are the F2 progeny we are getting by self-pollinations. First, you have to see the conditions to be remembered. So these are the conditions when capital P and capital I genes both are present. So the green color will be obtained. Why? Because capital I gene suppresses the or inhibits the expression of purple leaf color. That's why green color phenotype will be produced. So the second situation is when small p is present with capital I gene, with capital I. Here you have placed either capital I or small I gene, but the phenotypic expression is green color leaves. Why? Because here there is no capital P, so small p meant for green color. Okay, so the third situation when two genes with this in recessive condition, they ultimately produce green color as the parent. Okay, so when the capital P present with the small I gene, here there is absence of capital I gene. That's why then only they will produce purple color. Now we will see the punnet board. So now we will understand it easily. Okay, look at the Punnett board. So whenever you find capital P gene with capital I, then they will produce, they are going to produce green color leaves. Why? Because capital I is the inhibitory gene for purple color. Okay, so here you are getting 13 green color progeny, only you are getting 3 progeny with purple leaf color. Why? Because 3 situations are there. There is absence of capital I. So here you can see capital I is absent. Here also you can see the capital I is absent. Here also you can see the capital I is absent. So here we are going to obtaining the ratio of 13:3. So 13 progeny produced with a green color and 3 only obtaining purple colored progeny. Okay, let's see the next topic, nothing but duplicate genes. So the two dominant genes, either alone or in combination, produce same phenotypic expression. So when the two dominant genes, either alone or in combination, for example, capital A, capital A and capital B, those two are the genes. So capital A produce and B produce same phenotype. When they present alone or in combination, they are producing same phenotypic expression. So recessive forms of both the genes when together produce different phenotypic effect. So small a, and small b, when present together, they produce different type of phenotypic effect. Okay, so let's see the example for this. Fruit shape in shepherd's purse. So that is the example for duplicate genes. So interaction of duplicating genes was reported by G.H. Shell in the year 1914 in the plant Capsella bursa pastoris. The common name of this plant is shepherd's purse. Capsella bursa pastoris. Okay, so interaction of duplicating genes was reported by G.H. Shell in the year 1914 in the plant shepherd's purse. Let's see the cross. So in shepherd's purse, the presence of either gene A or gene B or both together, that means together, results in triangular capsules. That means capital A gene producing triangular capsule, capital B gene producing triangular capsules. They together produce triangular capsules. When both the genes are in recessive condition, that means small a, small b condition, they will produce oval capsules. Okay, so let's see the cross. Let's see the F1. Let's see the cross between triangular capsule plant with capital A, capital A, capital B genotype crossed with oval capsule plant, shepherd's purse plant with the genotype of small a, small b. Okay, so capital A, capital B, capital A, capital A, capital B, the phenotype is triangular capsule. Then they are present in recessive condition, they will produce oval capsule. When they are crossed, okay, F1 individuals are obtained. F1 individuals are also having triangular capsule. That means when the two genes present alone or together, they produce triangular capsule. But the recessive forms of these two genes, like parent, they produce oval capsules. Okay, so let's see the conditions to be remembered, such as when two genes present together, they produce triangular color. When only capital A present, also produce triangular triangular shape fruit shape, sorry, triangular shape fruit shape, when only capital B present, also produce triangular fruit shape. When both the genes are absent, that means the recessive forms of two genes producing oval capsules. Oval capsules. Okay. So let's see the Punnett board. So these are the male gametes are produced from male parent. Okay, that means F1 individual is going to self pollination. When F1 individuals are allowed to self-pollination, we are going to get the F2 progeny. So before going to F2 progeny, you have to be remembered the situations to be remembered. These are the situations. When both the genes are present, they will produce triangular capsule. When only one capital gene is present, then also triangular capsule produced. When another capital B gene is present, then also it will produce triangular capsule. When both the capital genes are absent, that means both are recessive genes are present, then only it will produce oval shaped capsule in Capsula bursa pastoris. Okay, so let's see the Punnett board. When F1 individuals are allowed to self-pollination, so these are the combinations we are going to get in F2 progeny. Okay, so when both are present, it will produce triangular shape. Okay, have a look. When only one capital gene is present, then also triangular shape produced. When only capital A gene is present, then also triangular shape is produced. The oval shape obtained only one situation is there. Both the genes are in recessive condition, then only oval shaped capsules were obtained. So the phenotypic ratio is 15:1. 15 triangular capsules obtained, only one oval capsule is obtained. Okay. Let's see the third one, that is polymorphic gene. Polymorphic gene. Okay, here the two non-allelic genes controls single character. Two non-allelic genes controls single character. That means single character is controlled by two non-allelic genes. They produce identical or same phenotype when they are alone. Two genes are going to produce same phenotype when they are alone. So when both the genes are present together, their phenotypic effect is enhanced due to cumulative effect. When the two genes are present in single individual, the phenotypic effect is enhanced due to the cumulative effect of those two genes. Okay, so let's see the example. On's length in barley. On's length in barley. On is nothing but I will show the picture. On is nothing but a fiber like or bristle like structure present on the head of barley. Okay, so look at the picture for better understanding. So these are the awns. Okay, so now coming to the example, that is on length in barley. On length in barley, that is example for polymorphic gene interaction. Okay, so when two dominant genes such as capital A, capital B, when two dominant genes capital A, capital B alone gives rise to awns of medium length, but when both are present, long awn is produced. Why? Because cumulative effect of A and B that enhance the phenotypic expression. When they present alone, they produce medium length on. When they together produce long on due to cumulative effect of A and B. Okay. So absence of both results in onless. When both the genes are absent, the seeds are produced onless. Okay. So head is not having ons. Okay. So let's say that cross, when pure breeding long on plant with a genotype capital A, capital B, capital A, capital A, capital B, capital B is crossed with on less plant, nothing but small a, small b parents. So these are the gametes going to produce when they fuse in to produce F1 individual which is having long on. Okay, which is having long on. Why? Because both the genes are present. That's why we are getting long on as parent. Okay. So when they allowed to self-pollination, we are going to get F2 progeny. Let's see the Punnett board. So this is the Punnett board. We are getting in F2. These are the F2 ratios. Before seeing the Punnett board, you have to be remembered these situations, such as when both the genes are present, cumulative effect. Due to cumulative effect, long on is produced. When they produce alone, they produce medium on. When both the genes are absent, they produce onless. Okay, so now we are going to see the Punnett board. So let's see the Punnett board. These are the gametes, male gametes and female gametes going to fuse in these combinations. Then we are getting 16 types of progeny in F2 generation. So wherever you find capital A with capital B, long on is produced due to cumulative effect of these two genes. Whenever you find either A or B alone, they produce medium on. Okay. So if both the genes are absent, they produce onless. Okay. They produce onless. Okay. So this is the phenotypic ratio we are going to get in F2, that is 9:6:1. 9 are long on having both the genes. That's why cumulative effect of these two genes producing long on. If any one gene present, either A or B present, then they produce medium on. If both the genes are absent, that means both the capital genes are absent, both are in recessive condition, onless is produced. So the 9:6:1 ratio, 9 long on, 6 medium on, 1 onless is the phenotypic ratio of polymorphic gene. Okay.